So it's a great pleasure to introduce Todd Golde, who's our speaker at the Shiley-Marcos ADRC seminar this morning. I will keep Todd's introduction as short as possible so I give him enough time to be able to complete his talk. So Todd obtained an MD PhD, trained at Case Western, and did some great research with Steve Youngkin looking at amyloid beta protein, completed neuropathology clinical chemistry training at University of Pennsylvania, did a lot of great research at Mayo Clinic in Florida, and most recently he has moved to University of Florida. He's currently a professor in the Department of Neurosciences at University of Florida in Gainesville. He is the director of the McKnight Brain Institute and also the director and PI of the One Florida Alzheimer's Disease Research Center. And he also heads the University of Florida Center for Translational Research in Neurodegenerative Diseases. Todd has authored almost 300 papers on a variety of topics. In addition to amyloid beta protein and Alzheimer's, he has contributed to many efforts on translational research. To highlight a few, contributing to the first demonstration of gamma secretase modulators in 2000, and developing AAV vectors and slice culture models of disease, and more recently looking at omics and innate immunity as approaches to therapeutics. So without further ado, we welcome Todd, and he's going to tell us about some new twists on neurodegenerative disorders.
Oh, thank you, Doug, and everybody. You know, it's good to see a lot of friends and new people. If people have, you know, I like to keep these informal. It's hard over Zoom, so please, you know, just shout at me if I'm, you know, if you want to interrupt. Okay, so I'm going to mostly what I'm going to talk about. First, Happy New Year to everybody. We hope that 2020 is fully in the rearview mirror sometime in the near future, and that we could normalize our lives. But, you know, I think there are some good things that have come out of it. And one is that I think some of the ways that we share information scientifically is you know, we've learned to be comfortable with Zoom, and in many ways I think it opens up opportunities for collaboration and long distance things in ways that we hadn't really done as well before because we weren't really forced to. So anyway, I'm going to talk a little bit today about sort of the first part of the talk is really this new twist on an old hypothesis, and talking about our omic data that we've been generating in this collaborative U01, and how a biologist views it, not a bioinformatician views it. And then I'll go on to a few other things. Briefly, I have some disclosures. I'm a co-founder of a couple companies on their advisory boards, et cetera, and I have a number of patents related to AD therapeutics and RAV technologies. I'm not going to have time to lead the extensive list of collaborators that through, you know, this is only a partial list of those who have been through the years, but obviously the funding from the NIH and other places like Bright Focus have supported much of this work, but I'll try to mention the people who are really doing the work as I go along in the talk. So just a quick overview of what I'm going to talk about, a little intro just to get everybody on the same page, or at least so you could understand where I'm coming from. Then talking about these multi-omic studies that I think, if you view them in the right way, begin to give us some new insights into Alzheimer's disease, and also can place them in context to other neurodegenerative diseases. I'll talk a little bit about what Doug mentioned with these brain slice cultures of tauopathy and synucleinopathy, and then really this final point is if I get to it, we'll be trying to think about, and you know, I've had the pleasure of co-authoring some things with Doug over the years about thinking a little differently about AD therapies. So part one is, what can multi-omic studies in mouse models and human neurodegenerative diseases tell us about AD pathogenesis and possibly inform therapeutic targets. And this is really the U01 collaborative team, and a large number of people are involved, but Nilofer Ertekintanen and Nathan Price were my co-investigators in this initial foray. Nathan still left ISB, and now Corey Funk has taken over his role. But obviously the ADRC of Dennis Dixon Brain Bank had a huge role in this because most of the samples came from there. So basically, when we went into this now some 7 or 8 years ago, our focus was to try to provide a transcriptomic and map of that really leveraged the ATN framework for Alzheimer's disease. So basically, we took AD, PSP, what we call, or Dennis called path aging, others call high plaque controls, or divergent or discordant AD, where you have A beta, no tau, and no evidence for neurodegeneration or cognitive impairment, and controls. And we only focused on 2 regions, temporal cortex and cerebellar cortex. Cerebellar cortex turns out to be incredibly interesting. I don't think we could view it as a control tissue, which I'll try to make the point. And basically, we used a similar set of mouse models over a longitudinal data set to provide a sort of an assessment so we could compare what's happening in the mouse models, which are only models of AD relevant pathologies. And you could really argue that the tau mice are really models of frontal temporal pathologies. And ultimately, the goal of this transcriptomic exercise or proteomic exercise from the NIH point of view was to get to new targets. And I think that's going to be very challenging, but I think there are some things that can emerge from this and will emerge. So I began to look at this data, and I'm not a bioinformatician, and I'm going to show some plots, and I'm going to organize these in the first section in a way that's very different from what people are typically see. And so what I've plotted on the x-axis here is the transcriptomic beta. So this is similar to a log fold change, a log 2 fold change, but it's really not. But that's a good way to think of it. It's just the difference between AD and control. And this is on the left, or the y-axis is PSP versus control. So when we look at the temporal cortex, what you could see is that there's a positive correlation across all the genes above a threshold of, I think, a CQN value of -1 here. But as soon as you put any significance on the AD versus control data, you see that there is a striking positive correlation between the changes that you observe in the temporal cortex of AD and PSP versus their respective same set of controls. So in other words, the transcriptomic changes that we see when somebody dies with PSP or AD are highly, highly similar. Notably, in the cerebellum, you see the same thing. And one of the striking things was that just in the AD versus control cerebellar cortex, which basically most people would look at and say, it's not really being affected, at least certainly not visibly pathologically or any cerebellar symptoms that would be typically associated with AD. Again, you see this striking positive correlation. Notably, when this is just using a simple model adjusting for most covariates, but not for cell type. What in our comprehensive model shown below, you can see that when we adjust and normalize this data for using sort of candidates, cell type selective genes to adjust for changes in cell type, even the entire data set shows of all genes shows a highly positive correlation. And basically all of these are, some ridiculous p-value. This is just simple linear regression. So the bottom line is that whatever we're seeing in a transcriptome of somebody who dies with AD and PSP looks pretty similar in 2 regions of AD. So whether you make whatever network model you make or whatever, it probably applies to both of these. I think the even more interesting aspect of this is, and somewhat surprising, is when we took the AD temporal cortex versus control on the x-axis and plotted it versus the same set of genes in the cerebellum that were, and what we can see is again is a striking positive correlation. And as soon as you put any filter for significance on these for the individual DEG, you get a striking positive correlation. So basically the same set of genes for the almost, like 90+% of them are moving in the same direction in the temporal cortex as they are in the cerebellar cortex in AD, and that's the same as well in PSP. And so there are a couple things that I think that I wrote it out here that we've typically viewed the cerebellar cortex in human AD as unaffected. I think at least at the transcript level, it is not. And in fact, it's showing the same changes in gene expression that you're seeing in the temporal cortex, but it's just, not has visible signs of pathology there. So one could ask, well, what are these changes, whether you're looking at, changes that are common between PSP and AD or changes that are conserved across the two brain regions? And they're sort of what you might expect. I mean, there's a long laundry list of sort of geo terms that you could put this in. But for example, things that are going down in both are a lot of things related to DNA repair and mitochondria or protein glycosylation. And in the things going up that are shared are a lot of things that have to do with, again, DNA organization or developmental processes or metabolic processes. So I think it's really complex, and we sort of all know this. I mean, there's not only degenerative changes, but there are obviously compensatory changes that occur across these diseases. But I think when we're looking, at least at this stage of disease, what we're really saying is not causal, it's the consequences of neurodegeneration. So when we think about this in context of trying to find new therapies, I think we have to be very cautious. We're looking at the downstream changes, and it's going to be hard to winch back. Now, this is a little different. What we've plotted here is, again, AD versus control in the temporal cortex, but this is that same set of genes now, but on the y-axis is path aging versus control. And what's really striking about this, as opposed to having a positive correlation as we saw with PSP, there's a negative correlation. It's not quite as robust in terms of the R-squareds and the slope, but what it's really saying is that at least in the path aging cases that we've looked at, there appears to be a completely almost inverse change in the gene expression patterns. So it's almost like if we presume that these people are going on to get AD, that they're just in this A stage of AD, then it's saying that that initial change is actually in the opposite direction for many of the differential expressed genes. And so we think we've corrected this with the second batch of ADs, but this is, of path aging cases, but in this initial case, there is a caveat that path aging versus control, few of the changes really withstand a false discovery rate correction, even though the AD versus PA values are highly significant. They're not necessarily significant between path aging and control. The initial series had fewer cases of path aging, etc. And I'll get into a little bit about what these genes are as we go on. So this is a little different way of looking at this transcript data, and there's a lot of data here. But I'm going to walk through it, and I'm going to show you a series of slides that are all the same in terms of the order of what's on them. And each dot on any of these slides represents a single differentially expressed gene, and the beta or log2, our pipeline for assessing the mouse data, was a little different than it was for the humans. So, but basically you have the temporal cortex data here, where we have the changes in AD versus control, path aging versus control, or PSP versus control. In green is the cerebellar data, again in the same order, AD, PA, PSP. And then in orange is a APP CRND8 mouse that gets amyloid deposition beginning at about 2 months of age, and we analyzed it at 3, 6, 12, and 20 months of age versus control. And so what you can see is if we filter on a set of genes, let's say, that are up in AD versus path aging, you see that is maintained between the cerebellum and AD. But what's interesting is if we look at genes that go down at 6 months in the CRND8 mice, we can see that a lot of these genes sort of follow the same temporal pattern. They're down, down, and then as the mice age, they normalize, not dissimilar to what we might hypothetically propose as you go from A to T and N, and from going from path aging to AD. So just to show that there's some validity to this that I think that everybody would accept, what we've looked at here, and I could take any microglial set of genes almost for this that are significantly altered. I just chose to focus on one that was recently published from Bart and Strupper, where they did spatial transcriptomics to identify a set of genes that are upregulated in microglia around plaques, or microglia and astrocytes, one should say. And one can see if they look on the right side that of this graph, that as the mice age, all of these are induced, and for the most part, they're up in our AD versus control series, but they're really not changed in path aging too much or in PSP too much in the temporal cortex. And there is some slight trends towards elevation in the cerebellum. So again, giving some face validity that looking at an immune system pattern, there is, we're seeing something that's relevant to other data sets. But then we could focus on just, again, what are some of these genes that seem to be downregulated in path aging and upregulated AD, and those that are reflected in mice. And what begins to emerge is you could look at these genes and show that they are, in this case, a lot of these are endothelial genes. So this is just from Ben Barr's data showing a candidate one here, PTRB, and I don't know why I'm getting green lines on my screen, but what you can see is that these are endothelial enriched transcripts. So it's sort of interesting that in this our mouse models, which are models of amyloid, not tau or not neurodegeneration, show a downregulation of endothelial transcripts early that then sort of alters, and we sort of see that same reflection in AD. And I'm going to try to get rid of that. I'll just do a laser pointer. Maybe that will work.
It could be a host that's got marking on. So I'm going to ask whoever's the host to try to remove the markings.
Okay. And I'm not going to belabor this, but basically another group of genes that are found in this module, or what I call a different way of looking at a module, is also our oligodendrocyte specific genes as well. So genes that are highly enriched for expression in oligodendrocytes, not in other cells, or down in path aging, seem to be actually increased in AD. And they follow a similar trend of, they don't necessarily increase on average in the mouse model, but they do go down and then up. Notably, what's sort of interesting about this is when one reads into the literature on transcriptomics, the genes involved in active myelination or remyelination are often cholesterol synthesis genes. And I never quite understood or had a good biological explanation for why we saw these largely being upregulated in our mouse model of also, our mouse model of amyloid deposition. But it's clear that these are upregulated early, and it may be pointing to the fact that there's some damage occurring to oligodendrocytes, which others have pointed out potentially occurs, but occurs apparently with some sort of sequencing of events. But this is sort of a lifelong elevation of these genes involved in cholesterol synthesis, and they're really not consistently changed in the human samples. And then a final slide on this just shows that, well, what are these genes that are up in path aging and down in AD? And when you look at those, they're actually genes that are highly enriched based on geo-ontology for basically their neuronal genes, especially neuronal genes that encode genes that have a role at the synapse or in neurotransmitter signaling. And I really find this data fascinating because it sort of says that there's an initial response to amyloid in the human brain that's opposite of what we see as the disease progresses. And one will note that in this case, as opposed to some of those other sets of genes I've shown you, this really isn't reflected in our mouse model. Our mouse model actually shows synaptic gene downregulation early, and then it sort of normalizes as they move on. So I think it says that, you know, we could take a picture of a snapshot of our mouse models at any point during time, and you could make a story about how it does or does not reflect the human disease. But having a longitudinal data set is important. And the best we could do in humans is to have these discrete categories. But I think there may be something here that suggests that initial responses to amyloid in the human brain are different than what we see at the end stage, and that we could learn something by trying to understand the concordance or discordance via our mouse models using this sort of informed view. I'll just finally make the point that unlike in mouse models where we could clearly show, you know, the red, for example, are just the changes in the values for TREM2 or CST7, two immune genes that in microglial selective genes we're pretty familiar with. And you can see that as the mice age, you get clear separation between the amyloid depositing transgenic mice and their non-transgenic litter mates, even though there are changes with age in the non-transgenics. But in humans, this never happens. There is no single transcript, no matter what transcript you look at, that clearly distinguishes human from AD from control. There's just a lot of variance. And I think you have to be really cautious when people, especially when you look at the scatter in humans, when somebody publishes on 12 versus 12, and even if it's, you know, especially when it's single RNA, they can make up whatever story they want. If they happen to pick 7 AD cases that were sort of in the high range and 5 that weren't versus some controls that were down, I just think it's, you know, these studies need to be well powered in order to do this. So that's all I'm going to talk about for the most part on the transcriptomics. I think it's provocative and interesting to think of, especially with this temporality. But I think more people will find what we've done now in the proteomics and trying to actually, in this case, it's sort of been a crosstalk between Alan Levy, Nick Seyfried, and folks at Emory and our group, going back and forth between the mouse models and the human data. And I think this, to me, is more exciting and could give more traction on what's going on in the disease. So what I'm going to focus on today is a highly significant module that emerged from the human studies. And in parallel, we were seeing something very similar in mice. And so you'll see the set of genes is some of them you may have heard of in the context of disease. And I'll put a little, I'm not going to go through listing each one. It will just take too long, but you'll hear about these in the future as I go through. But in this particular module, correlates well with clinical status, amyloid, and tau, and they call it module 42. But this is now looking at our CRND8 mice between 12 and 20 months old, doing proteomics on the same platform, TMT proteomics that they did at Emory. And what you'll see is this M42 module has almost complete overlap between the human and mice. I mean, it's 30 out of 32 in the module. And those 30 that overlap are highlighted in red here. And one of the things that's important is to just put in context, this is p-value here and log fold change. And you can see things like APP driven by A beta, ApoE, clusterin, alpha-1 antichymotrypsin, serpent A3N in mice, you know, are all things that we know are accumulate in the AD brain and are in many cases, at least clusterin and ApoE and APP are genetically implicated. But these other proteins are accumulating based on this unbiased proteomics or less biased proteomics to nearly the same degree. And there are other genes in here that I'm not going to highlight too much. A lot of these are microglial genes or astrocytic genes, or in many cases, we don't really know what they're doing, or proteins, I should say, because this is proteins. But we sort of made a hypothesis based on this when we plotted the RNA values or changes in RNA versus the changes in protein. And what I just want to say was it was able to put some biological framework that we've actually been not too bad about our guesses here, that if things are not changing very much at the transcript level, but are accumulating and appear to be neuronal, they likely end up being in dystrophic neurites. Proteins that are the most elevated tend to be accumulating in plaques, and a lot of these that change quite a bit on the transcript, but not at the protein level, are often immune or microglial. And I'll show that point in these next two slides, where this is the mouse data. We don't have the same kind of data yet for humans, but what we see is that you could have many transcripts that increase almost fourfold or more, and there are things like TREM2, but they're increased 15 or 20% at the protein level. Further, we have many immune transcripts that are upregulated dramatically and really aren't changed, at least at first pass in this proteomic analysis of the protein. So this is suggesting that by having both the proteome and transcriptome data from the same cohorts or the same model is really important to give you an understanding of what's going on, because to me, this looks like there's a big disconnect, especially in immune molecules, between what's happening at the transcript level and the protein level. And I think that's going to be important to understand in the future. We need to validate all of this. We're still in the early stages of this, and it involves, painstaking work of going through and finding antibodies that you really trust. And we're in the process of doing that. And so far, more often than not, we validate rather than invalidate this data. I think there's a really important paper for people who are interested in this from Jan's group up in Canada, Bouteille et al. And Cell Reports, where they show, following an LPS challenge using a ribotag approach, that there are huge disconnects between the transcriptome and proteome in microglia specifically, that are very similar to what we're witnessing here in our APP mice. And I think this probably may be one of the more important insights we gather from these modeling studies about what's going on in the immune system. And to me, it looks like a very strong interferon-like signaling component, but I don't think it's driven by interferon gamma, perhaps per se in the brain, because we just don't see interferon gamma expressed at a level that would be consistent with driving this. It must be some other crosstalk with this pathway that's likely resulting in translational repression. So again, based on this sort of omic data, I tried to reduce this to some testable hypothesis. And the idea was that by understanding what's happening to the protein and the RNA, we could bin these and inferring what cell types these may be being expressed, and we could begin to understand where these are accumulating in the AD brain in our mouse models. And just again, notably, 3 of our top increased proteins that, besides clusterin and ApoE, had previously been shown by others to be increasing or be plaque associated: midkine, pleiotropin, and vitronectin, either by Tom Wisniewski, sorry, not Henry, and Pat McGeer, and others had looked at these, and then others had also been implicated, like collagen 25A1 is the AMY epitope that was originally found by Virginia and John as a plaque-associated protein. This is probably out of place. This paper is now in press. Another reason that there could be a disconnect between our models and in terms of the transcript and proteome is intron retention. And we've just looked at our data and seen that there is quite a bit of intron retention, both in the AD brain relative to control, as well as in the mouse models. And interestingly, in the mouse models, a lot of the intron retention events are occurring in the mouse in what we infer are microglial selectively expressed genes. So again, in this last part, I'm going to, of the omics part of this, I'm going to just focus a little bit on a few of these proteins, midkine, pleiotropin, and I'll show a few other data on these, and then I'll try to pull it all together here in a minute. But if you look at, this could be midkine and pleiotropin, and I'll show some additional stains of these. If you look at these and take antibodies to these that we validated know that they're specific for the target. It looks like you're staining amyloid. Vitronectin also looks that way, although it's also seen in astrocytes. But we also see other proteins. Some of you may be familiar with HTRA1 because it's a cause of familial forms of CADASIL in addition to Notch, so mutations in this. But in this case, it looks like a very good marker of dystrophic neurites. And I'll show some better pictures of these as I go on. But I just want to, we decided before we got too far into this to do a little pilot study using our AAV expression models to ask, well, pleiotrophin and midkine appear to accumulate in plaques in humans and in mice. There have been some work done on them. And we decided to ask whether overexpression of these would alter amyloid deposition. And pleiotrophin and midkine have really complex signaling. They bind a whole bunch of overlapping receptors. And so trying to knock out a receptor or understand the receptor signaling we felt was going to be challenging. So we just chose to overexpress these. And somebody has their phone on, so I'm not sure. I'm getting a little bit of background. And again, we're in the process of reproducing these data in a second cohort of mice that obviously was delayed by COVID. But what we do see is that pleiotrophin, when overexpressed, reproducibly increases amyloid deposition at 3 months of age. So we inject these mice at P0, wait till 3 months, look, they increase amyloid deposition. But more strikingly, with both pleiotrophin and midkine suffers from having a little more variable expression than we would like. We see that at 3 months of age, we've never observed amyloid deposition in the vessels, or CAA, in this mouse model. It takes typically till 9 or 12 months before there's appreciable CAA in this model. But there was a striking redistribution of in the pleiotrophin mice, as well as in some of the midkine expressing mice, of amyloid into the vasculature. And we've never, I've done a lot of manipulations in this mouse model. We've never seen anything like this. Further, we had tagged these proteins with an epitope tag so we could see where they're being expressed. And you can see that it's mostly in neurons in this case. But these are small plaques. I don't want to belabor this. You just need to trust me a little bit. I don't have all the controls on this slide to show. But these are small nascent plaques in the 3-month-old mice. And you can see exquisite colocalization of midkine and pleiotrophin. So this is also sort of cool because by overexpressing these, we could actually sort of confirm our observations that these are proteins that accumulate in plaques. And again, we've gone on, this is work done with Jorge Trillo Lopez, Tony Agnes, and Stefan Prokop. And one could see that if you look at midkine, this is using a commercial antibody, and I could give you the name of it if people would like, but basically it looks like amyloid. It seems to be more specific for compact amyloid. But if somebody showed, again, this picture to you, you'd go, oh, that's amyloid. And so this is amyloid on the bottom, and this is, the serial section stained with, or a section from the same brain stained with a midkine antibody. Further, we could look at this in mice. This is a different antibody that works better in mice, but the green is thioflavin S, the pink is the midkine, and you could see that what's really sort of cool is it's very, very specific for co-deposition in what appears to be the core of the plaque in the mouse models. So we think this is pretty cool. And we've been buying a lot of antibodies to this list of proteins that we think are possibly novel plaque or amyloid associated proteins, as we call them. And a lot of these just didn't work. And so we went back to the drawing board during COVID and made a lot of antibodies that were polyclonal that would hopefully both recognize the human and mouse forms. Not all of these work, as everybody knows, but a lot of them did. And we can see, for example, here's pleiotrophin. This is all data in mice. Much of this is reflected similarly in humans. Here's a protein called EGFL8, so a member of the EGF family that appears to be plaque specific. Olfactomedin-like 3, again, plaque specific. Things like ApoD that had been implicated as being a plaque associated or amyloid associated protein before. Eliezer, back when he was at UCSD, actually had already come across BMP6 as a very good marker of dystrophic neurites. And obviously we have several of these that do this. And then you have others that seem to be both in dystrophic neurites as well as binding to the plaque. So what do we think's going on here? Well, if you look at this list of proteins, some of which we've now confirmed as truly being in the plaque and others seem to be in dystrophic neurites, what one can see is that many of these are, if not most, are thought to bind heparin or heparin sulfate proteoglycans. So sort of back to the future here. And we know that heparin sulfate epitopes are increased in plaques as well as the heparin sulfate proteoglycans themselves. And we know that many of these are molecules that on this list are potent signaling molecules. And so in addition to clusterin and ApoE, which we know are well-established amyloid-associated proteins that have also effects on amyloid deposition itself. So again, this is sort of the title of the talk. What do we think may be going on here? Is that, well, maybe, the disconnect between amyloid deposition and the neurodegenerative disease is not because amyloid is a direct neurotoxin. I mean, that's the hypothesis we've been sort of under for the last 25, 30 years. And people have been trying to find the potent toxic form of amyloid. But it's pretty clear that these other proteins that co-accumulate in plaques not only can modulate amyloid deposition, but have biological activities of their own. So these models aren't necessarily mutually exclusive, but what about the possibility that the co-accumulation of these other proteins in the plaques are key mediators of the downstream phenotypes? And I think that's the hypothesis that we've sort of settled on as to test and see if it's occurring. And we also think that this notion that heparin sulfate content, and of course with the ApoE Christchurch, that's reemerged as the forefront, may be very important in understanding the role of these amyloid-associated proteins and their role in AD pathogenesis. So just to quickly summarize, I showed you about a shared transcriptional profiles between AD and PSP, and also a shared transcriptional profile across a severely affected region of the AD brain and an unaffected AD brain. And I've sort of used this to galvanize the thought in my head that, if we don't start thinking of symptomatic AD as brain organ failure and devise treatment strategies appropriately, I think we're going to continue to fail. There's so much going on in these brains. It's so complex. We need to start treating it as if it is an organ failure and attack multiple aspects of the pathology simultaneously if we're going to significantly move the needle. I've talked about the disconnects between the proteome and transcriptome that need to be validated, but I think they're telling us something important about the pathobiology of AD, or at least our mouse models. And I just like to then say, I think when we think of our mouse models of amyloid pathology, they're pretty damn good. At least using unbiased proteomics, they're highly overlapping with what's happening in the human AD brain. There are going to be some disconnects for sure, but I think it's really important that when we're viewed in the appropriate context, they could be, this could be important. And then I think ultimately we need to think more broadly about these, sorry for the typo, amyloid-associated proteins and their impact on disease. And I think I've given you some evidence for why we need to do that. I'm going to quickly run through this next part of the talk. We've published most of this, but I'd like to acknowledge Kara Croft, who's a postdoc in my lab, who's a UK dementia fellow and going back to Britain shortly, and she drove all this work and made it happen with a lot of help from others. It's taking advantage of our AAV toolkit. I don't know if anybody on this call has used some of our AAV-based technologies, but we've really developed quite a toolkit that enables us to do a lot of things quickly and more rapidly than one through triple transgenesis, and perhaps one day translate this into gene therapies for humans. And I won't go through this, but to say if you want to do something in the neurodegenerative disease space using AAV, come talk to me because we probably got some tools that will help you and we're happy to share them. And again, that toolkit was built by Pedro and really sort of cool, a large, diverse group of undergrads who over the last 10 years have come through the lab and helped Pedro to build this. And it's really an amazing toolkit. So Cara came into the lab and had worked in Wendy Noble's group on brain slice cultures of mouse models of tauopathy. And the first thing we did was just see if we could transduce these brain slice cultures. So we take them at P8 or 9. They're 350 microns thick, and they're grown on a millipore membrane at the air media interface. And we could keep these alive for almost a year in culture unless they get contaminated. And notably, all I'm showing here that's important is we could transduce whatever the AAV vector will get into, neurons specifically, importantly, microglia selectively, oligos or oligoprogenitor cells or astrocytes. And indeed, it's the most robust place for AAV transduction of microglia that we've seen, which I think is important for the field. And though the microglia start off being sort of activated and ramified because they're cleaning up the debris in the slice culture, they seem to quiesce and assume a more homeostatic state as they go on. But we could routinely get 40 to 60% of these transduced in the culture, and that's the best situation we can. Kara in this initial paper was able to show that we could drive tauopathy and synucleinopathy in these cultures in short periods of time. So this is 2 months, and actually this is only just 1 or 2 months. So this is just 1 month in culture. This is thioflavin S positivity. This is using a combination mutant of 2 FTD mutants, and there's something special about the S320F mutant. We don't know what it is, but it really rapidly forms tangle pathology. It will do it by itself. P through an L will also go on to form tangles at about 5 to 6 months by itself. I don't show that, and it's not published yet. But every one of these cells that's transduced is now got a bona fide neurofibrillary inclusion or tau inclusion by immuno-EM and thioflavin positivity. The synuclein inclusion seemed to be more like a pre-Lewy body. If we actually seed them with exogenous seeds, we also can induce what appears to be a more mature looking Lewy body. But again, very rapid. We didn't pursue this as much because we don't, like just in the transgenics, typically there's very little difference between wild type and the mutants, whereas here wild type would serve as a nice control. So what I'm going to show you in the next few minutes is quickly what one cool observation that we've been able to make using this. And that was addressing the question of, do inclusions turn over? And for this, we used what we call an optical pulse chase, where we made dendra. So dendra is a GFP mutant that can be photoconverted so that it irreversibly switches fluorescence from green to red. So the first thing we did, and I'm not going to go through this in any detail, is just confirm that the dendra versions of these molecules behaved like the untagged versions that we previously published on. And the bottom line is, yes, they do. And even the dendra 2X, as we call it, P through an L S320F mutant forms true neurofibrillary tangles. So what we do in this is we transduce the slices at day 0. Right after we make them, we wait 4 hours, change the media, add virus. That's important for those who want to reproduce this. And then we wait till 10 days. We've now extended this, and then image and convert. So you shine blue light on it. You have to be careful. If you shine too much, you could kill the cells. You don't want to do that. And then we image them, the same region, over and over and over again. And what you can see is the dendra converts to red and goes away. Wild type tau converts to red and goes away. The cells are still alive because they're expressing green fluorescent protein and continue to do so. But you can see that even in the P through an L S320F tau, which is forming tangles, about 80% of these, even at 10 days in vitro, we feel are true tangle forming based on thioflavin S data. Not thioflavin S, but thiazine red in this experiment. And you can see that these persist, and that is reflected here, but it's better appreciated when we look at individual cells, where we can see that the half-life of these tangles is tangle-bearing cells is extended to about 7 days on average, even though there's a wide display. And notably, we see sort of different patterns, but the most important one is that the red begins, is accumulating, goes away, and it's replaced by green. And in most cases, there's not a huge amount of growth of the amount of volume in the cell or area in the cell that's replaced by fluorescence. Notably, as we go on, we can see that the tau inclusions, if we wait to photoconvert until later in times in culture, so 30 or 60 days, we get a corresponding increase in the half-life of the tangle-bearing cells, while there's little change in the wild-type tau or dendra-bearing cells. So if we wait till about a month in culture convert, you see the half-life now on average is extended out to about 15 days, and then it increases further out to about 20 days. So as these cultures age, for whatever reason, is it the age of the tangle or is it the age of the culture? We don't know. There appears to be less rapid turnover. But nevertheless, the point is tangles are not, at least in this newly formed tangles, at least in this organotypic brain slice model where all the appropriate cells are there, are turning over, at least based on this evidence. We can also seed tau using addition of tau fibrils in the Virginia Lee Mark Diamond experiment. This worked incredibly well in these slice cultures, and we could show exactly the same thing, and we could show that aging of these cultures produces an extension of half-life as well. So I think if fibrillar tau inclusions turn over in the time frame we are observing, then this means that we may be able to clear tangles if we really understand these mechanisms. These inclusions are not tombstones. The tag versions, though fibrillar by EM, they're still tag versions, though they're fibrillar by EM, so that does raise some questions. It doesn't really explain the new tau incorporation. It does still rely on dendra fluorescence being really stable over these time periods, but I think the aging experiments give us more confidence that what we're seeing is real. And this aging effect is interesting, and we wonder whether there is a cap to it. I think this paper is now accepted, provisionally accepted in ACTA Neuropath, so hopefully it will be out pretty soon. And we're trying to follow this up using other methods to show that it's really real. And then I'm just going to show one quick video. This is CX3CR1 GFP brain slice, so you can see the microglia in it. And this was a mistake. We dumped A beta on the, I'm going to need to get out of my, turn the laser pointer off. If you focus on the slice, this is dumping fluorescently labeled A beta that's aggregated on it. You see that there's almost no accumulation of A beta. You have to, everything's happening here quickly. You're looking at seconds on the top. But you can see the microglia that have crawled off the slice, and you have to trust me that they're microglia. We've shown that. Accumulate A beta, and they get rid of it really clearly, at least what appears to be soluble A beta. But in this experiment, we happened to put some chunks of A beta on. It was a mistake. So it had aggregated on us. And what I'd like you to note is that the microglia in this slice take these chunks of A beta, especially off the slice, and they sort of compact it. And for those who were around in 1991 or whatever, when Henry Wisniewski, may rest in peace, got up and said, "You see, microglia are making A beta." Well, there may be something to that. At least they may play a role. I think we now recognize they play a big role in the disease. But I think one of the things this may be telling us is that if they can't eat it, what they do is they compact it. And we're pursuing this, but we'd also just like to say that we think that this model, where we have what we think are sort of dystrophic microglia that have crawled off the slice, where the relatively homeostatic ones in the slice may be very valuable and could help us to understand what's happening with microglia in the brain in a more accessible experimental environment. And I think then this just is showing that, for example, we could introduce a calcium sensor into microglia in this system. And again, very early data, but we could show calcium signaling in microglia. And so if we could use any kind of GFP labeled sensor, we could begin to study microglial functioning in the context of other cells that we know are important for keeping them in balance and in a more homeostatic state. So I think I'm just I'm gonna mention this and then stop and then take questions rather than going into the data. But Doug contributed to this work and Steve Dukoski, where we talked about this idea of, there's so many noise out there in the field about why therapies aren't working. And I think in many cases, one of the reasons is we've simply been doing the wrong drug at the wrong time. And when we have drugs that work, we continue to not, or hit their target at least. We need to really try to align the studies that we're doing in the clinic, all the challenges that are with prevention studies that arise, such as toxicities. We need to try to overcome them and continue to treat the disease in its pre-symptomatic or preclinical phase. And once we're in the symptomatic phase, I'm not particularly optimistic that unless we use a magic shotgun or a magic cocktail approach, that we're going to be very effective in treating the disease. Because we really, once we show symptoms, we're in at least regional brain organ failure. And it's going to take a lot to alter this. So I'd just like to say that, we think we could do this, at least proof of concept, with there's no even preclinical roadmap for this using our AAV vectors. And I'll just skip there and then talk about, if somebody wants to ask me about this, we think we have a pretty cool tool that's targeting the HPA axis that could be a type of magic shotgun in Alzheimer's disease that targets not only A-beta, tau, possibly neurodegeneration, but also has a broad organismal effect that could be beneficial. And again, this work has been done by lots of people, not me. I'm simply a conductor at this point, and I rely on great hands and great minds in the lab to make all this work go. And I didn't talk about the One Florida ADRC, which I direct. I think like many people, we've been on a holding pattern with our clinical studies, but I'd love to talk to Jim and Hector, later date and others involved to see how, our efforts in Hispanics and our focus on that population in the One Florida ADRC could align with things going on at UCSD, and perhaps we could find some enhanced synergies now that our funding, at least, we're through our initial funding period, and I think most of the hiccups with the transfer to the F30 or the P30 are done. So I'll stop there. Be happy to take questions and go from there. So one is from Doug to me: do pleiotrophin or midkind stain other misfolded protein inclusions? See no evidence for tau or synuclein. One of the interesting things is, like ApoE and clusterin, and heparan sulfates, do they stain peripheral amyloids? Haven't looked at that yet, but it's on our list to do. One of these actually turned out to be quite interesting. It was mostly in dystrophic neurites, but in humans it stained tangles, and we're still not confident it's our own antibody. We know it seems to be reasonably specific, but we're trying to get a second antibody before we pursue that further. So, but we have I think this data on the proteomics offers, as long as we're careful with our antibodies and make sure that we do, this will provide a great opportunity for many in the field to look at a whole bunch of new clinical pathological correlations. So fire away, people. Go for it. Thanks for your attention.
Anybody else? Questions? I'm sure there must be.
There are a lot. I'm sure there are tons of people. I was going to just stay quiet because I know there's a lot more biological folks than I am, but I'm very intrigued by your model of being able to look at tau turnover and possibly looking at factors that could increase turnover and the impact of aging, and whether you might be able to do this in maybe some of these iNeuron type, Rusty Gage type iPSCs.
So Matt Lavoie just joined us and has the iPSC technology. We've shown we could induce it in the iPSC, so we hadn't done the Dendra experiment, but that's certainly on the list. I think the real question is can we, as you say, the key is why does it turn over? how does it, I mean, the real, actually, I should give Brad Hyman's group some credit. I was sitting with Brad when his postdoc presented the data a few years ago on, tangles being function, or tangle-bearing cells being functional in a mouse model, or cows, able to. And I said, your data really sort of speaks to the fact that a cell handles a tangle, and I think we all recognize this. I mean, these tangle-bearing cells must exist in humans for many, many years. They're not a true prion. If if it was a true prion, it'd just keep on growing and blow the cell apart. So the cell has to compensate. So we're taking both a mechanistic sort of hypothesis-based approach. It's not easy because a lot of the stuff that we would try, you put a proteasome inhibitor on and it's too toxic. You put a lysis, autophagic thing on it, it's toxic. So we're using a genetic approach where we're trying to do in-cis Cas9 CRISPR of various targets, and then we're also using a unbiased approach to really look, because the temporal sequencing of the pathology development is so tight in this culture, we can, and actually we're doing that in primary neuronal cultures where we see the same thing, because we could get a higher transduction efficiency. We're doing omics on that. We're asking, what happens to the phosphoproteome, the proteome, and the transcriptome in a sequential fashion, and we could include all the appropriate controls, and maybe that will yield something. But right now, it's, we don't know what's turning over. But it's clearly the cell is adapting, or at least initially can adapt. The question, after time, do the tangles become less able to turn over? I think they must continue, because otherwise they keep growing, right?
Yeah, and I wonder if they're getting phosphorylated and becoming more resistant in some way. I don't know if you're able to look at that.
I had a question, Subhojit. Sorry, my camera is not working, so I was hesitant to ask questions. But I had a question about the amyloid-associated proteins, and I was wondering. So when you look, in the animal model, so I may have missed it, but the amyloid-associated proteins are also. Did you also see them in the animal model? Because I was wondering that.
So at least in this particular module, so that there is the overlap between the module identified in the human data on proteomics, and we still haven't gotten a complete concordance with antibodies that always work in the mouse model in humans.
Yeah.
We're working towards that, but pretty much none. There's been rare exceptions where we don't think the same thing is happening.
Yeah, no, I understand this. That's not my question. So I was actually thinking about the fact that if you just express the amyloid, in mice, then you do get the pathology, you do get the behavioral changes. So is it necessary for these amyloid-associated proteins to really be there for the amyloid to cause pathology?
Well, I think that a number of questions, the answer is our mouse models are models of amyloid deposition. They're not models of Alzheimer's disease. And,
sure.
I hate it when people call them that. They're not. So the question is if these, one is many of these proteins, at least temporarily, pleiotrophin and midkine are exceptions to this, are much more easily detected the older the mice are. So there seems to be a temporal staging to the accumulation in mice. We still don't know in humans. And so that's I think there's a lot of work to do, but it's saying, the first two that we looked at, I mean, there's been a huge amount of work on ApoE and clusterin, right? And ApoE and clusterin, the only difference between them and midkine and pleiotrophin, as far as I could see, except for that both midkine and pleiotrophin seem to influence CAA even more. Overexpression of these increases amyloid deposition, and they also, seem to be associated with the plaque pathology. So we have a laundry list of proteins that are accumulating, and I think that they're biologic. The question is, do they remain biologically active as they accumulate in the plaques? Do they create dystrophic signaling gradients? But viewing a plaque as, the field, we spent 30 years. It's not that, a lot, there's been incredible science spent on the reductionist theory that there is a toxic form of amyloid.
Right.
But maybe, I hate to the anti-amyloid people in the field, maybe there is something too, that amyloid is not the only thing, and it serves a role as a scaffold for other proteins to co-deposit.
I think the idea is very interesting. I was just asking about the specifics.
But I mean, all I would say is I was stunned at the overlap so far that we've seen between the human and mouse data. And the question is, what's different? I mean, again, I showed you some changes and similarities between the transcript and the proteome. I mean, the transcript in mice and humans, and some of those things are intriguing. I mean, I actually think maybe our mice are models of resilience, that the mice have a biological response to amyloid that enables them to do okay with it. Mm-hmm. And that we can learn from that response rather than viewing them as models of they deal with amyloid well enough, right? I mean, they're really not demented. Yeah. They're not the smartest mice in the world, but they don't have neurodegeneration. So something's different. And maybe it's just time. It just might be that there's not enough time.
Right. Thank you.
Todd, this is Ramesh.
Sure.
I like to learn your perspective on the PROTACs or targeted protein degradation, especially for undruggable targets. And I like to know a little bit about, any of these things. Probably, are you looking at it, where that particular tool is going to take us? Because I have seen several publications wherein some of these PROTACs are brain permeable and they are able to show some improvement. And I like to learn more from you, please.
Sure. So actually, a guy who's one of our better medicinal chemists in the College of Pharmacy here has developed many PROTACs or several PROTACs for cancer. And he reached out to us to collaborate, and obviously was delayed by COVID, but over the last couple months, we've developed he's made some novel PROTACs to different ubiquitin ligases. And we begin to see some really remarkable effects of these. We haven't yet put them into the slice cultures with Dendra. That work will start. But I'm encouraged. We've compared them to the published one from the eLife paper. I can't remember what group it was. And these seem to be quite better. That I could tell you, though, that in our initial experiments, just in cultured cells, we saw actually divergent effects, and we worry about I have some concern over specificity and toxicity over time, but I think it's a promising way, and it will be interesting to see if maybe that will converge on the biology of why tangles turn over. Maybe and that you can I think we have the tools now to ask questions like, if we have one of these agents, how are they affecting tau deposition? I mean, we got a lot of grief, and something that I didn't show today was one compound that's been marketed as it's an AstraZeneca compound that's a GSK3 beta inhibitor, and it was pulled because of toxicity issues before getting too far towards humans, but reproducibly lowers neurofibrillary tangle pathology in mice. And it's the only molecule that we've been able to reproducibly see effects on tangles in culture. And what we could show with that is it doesn't affect either synthesis or turnover of pre-deposited tangles. What seems to be it doing is preventing tangle formation in the first place. So again, I think having this tool will help us to provide some framework for how things are working. We could also do we only now established that we could block seeding with antibodies in culture, and we don't think that will have any impact on turnover, and we have some preliminary data that suggests that. But again, we could begin to look at this and ask, we have things like intrabodies we just published on it with some of the data that I skipped over. We have a molecular therapy paper where we show that either single chain antibodies targeting tau or intrabodies targeting tau can have effects on not only tau pathology, but modest effects on survival in a spinal cord model of tauopathy. but I think we got to do better before we have hope for these in the clinic. But again, we could ask, how are these really working? Because people really don't why would just having an intrabody that binds tau inside the cell really will it affect tangle dynamics, or will it only just prevent the conversion of a soluble tau to insoluble, and or can it clear a pre-existing tangle? I mean, we could begin to do some of that. And again, we're happy to provide these tools. They're especially, I think you could play this game with any proteinopathy relevant to neurodegeneration. I mean, you can make an AAV with it and put a dendra tag on it. You can begin to do some really cool stuff in periods of time with much, when you do in vivo multiphoton imaging, the number, people publish and they've done 50 events. We're looking at thousands of events typically in these studies, and you also have biochemistry that backs it up. So I think it's a pretty nice intermediate between the mouse model, some cell culture that doesn't have all the cell types in the brain, and what's going on in the brain.
Thank you, Todd. I'll be in touch with you soon.
Sure. Doug, you're muted.
I have a question since he's muted. So it's about that one slide you didn't really get into about the one agent with pleiotropic effects that impact AD. So I'm really interested in like pleiotropic genes that are influencing, metabolic and dysfunction and cardiovascular, and also AD in the Latino population, but also in like other people. And I'm just wondering, so this one agent that you're talking about, do you think it would influence people who have like obesity and Alzheimer's disease, people who have Alzheimer's disease and people who have obesity? Or is it, maybe something that's just because they have some sort of pathological mechanism, or would it be specifically targeted to people who have like obesity and that's right?
So, I mean, actually, I don't know if Rob's on the call, but, this is sort of built on some of the work Rob Rissman and others have done. And, there are labs at UCSD that work on stress pathways. So this antibody targets CRF. And the truth is came out of work where we showed that CRF antagonists really don't do what they should do to amyloid beta. And we said, we need to make an antibody to this. So we made an antibody. We got lucky. It was highly selective for CRF over the related urocortins. And where most of our really cool data is in metabolic areas. And the human data really is indirect, right? There's data that says cortisol levels in some studies correlate with cognitive decline. They correlate with amyloid loads in the ABLE, Australian imaging study. There's been long links of stress pathways to AD, but it's softer. It's not genetic, right? And I also think, this idea that, when we talk about, and the thing that attracts me to it has been the idea that in Cushing syndrome, where overactivation of the HPA axis typically through tumors or drugs, when you reverse that, you reverse the cognitive deficits, the depression, and even can reverse hippocampal atrophy. So, the truth is we're trying to get this, we're in the last stages of humanization of the antibody. We're trying to get funding to create the cell lines and make the antibody, but we're going to go into something like obesity as our first pass, because Alzheimer's disease is just too hard. But, if we find evidence that it is moving things in the right direction in the obese population, then I think it can be turned back to Alzheimer's. I just don't want to, but I think it's where we need to think. I think we need to really be careful about and treat, if we want to make an impact, again, later symptomatic stages of the disease, it's going to either take a little bit of luck. And the problem with a magic shotgun is you don't know if pellets are going to go off and have a bad effect as well, right? I mean, that's the problem with magic shotgun's approach, whether it's an HDAC or, what GLP-1 or whatever. It's just complex biology, and you only be able to test it in humans. I personally think we need a more targeted approach eventually for many neurodegenerative diseases, but I think we need to build a preclinical roadmap. I mean, there was Alzheimer's Association roundtable or conference on, combination therapies. And it was like a combination of failed therapies is likely to be a failed combination therapy unless there's really strong rationale based on, some biological data and hopefully preclinical data that could provide the rationale to move that forward. And yet we have there's almost nobody doing this. And trying to get funded on this, it's one of the frustrations. We have all this money in the Alzheimer's field, and they're trying to find, new causes of Alzheimer's disease. And I personally object to that. I object. I don't speak that loudly about it, but I think we know what causes this disease. The problem is, I just think intervening early is just so hard and requires such a safety measure that we need, it shouldn't mean that we should shut it all down, but it's going to take years to figure out what works in prevention and whether things are going to be safe enough. And unfortunately, things haven't worked yet. And I think, for those of us who have been in the field for, since in the early days of, since the early, the 1990s and stuff, I think we feel like, God, we thought we were just going to knock this. I mean, Wags will know. He was in industry. He's going, shit, we were going to knock this out of the park. It was just going to be smooth sailing. And it has been so humbling, I think, for everybody involved in therapeutic discovery. And, there are heroic efforts that are continue to go on through the work at UCSD and other places and the prevention initiatives. But we've also got to be smarter, because I think we have a window of about 5 years. And if we don't, people are going to say, why throw all this money? You guys are just wasting it. I do. I think we've got some pressure on us.
So unfortunately, I'm going to play the bad guy and say that we're out of time. If people have burning questions, you can perhaps address them to Todd through email. I'd like to thank you enormously, Todd. It's just a great pity that we can't go and do something in a major social fashion afterwards, but hopefully sometime later this year that will be possible. So thank you very much, and thanks everybody.